Good morning, everyone. After yesterday's wonderful lecture on perspectives that you develop in carrying out your research in microbiology by Dr. Kirti Dubey, the second lecture in the series will be delivered by Dr. Seema Nimbarte on the topic PCR and the diagnostics, molecular diagnostics application of PCR technology. And especially this is very important during the pandemic period. And it is also covered in your semester 6 curriculum. So we thought that to select this topic would be better to get the better insights of this topic. And we thought of Dr. Seema Nimbarte, who is an expert on this PCR technology. Now, let me introduce today's guest speaker, Dr. Seema Nimbarte. Dr. Seema Nimbarte is assistant professor in Sevadal Mahila Mahavidyalaya. She is MSc PhD in microbiology and is having 22 years of teaching experience and 12 years of research experience. Her area of specialization is applied and medical microbiology. She is alumnus of our department, and I remember her as a very bright student of the department. She is nodal officer of various BVOC courses in her college. She has published more than 15 research papers in the journals of repute, having very good impact factor. And she has published many videos based on your curriculum. And some of the videos that I keep sharing on the group is of Dr. Seema Nimbarte. She visited Singapore, Thailand, and presented her research work. Her specialization is in veterinary microbiology. She has completed 2 research projects which received UGC funding, and she was awarded 3 times Best Researcher. That means her posters were awarded Best Poster Presentation Awards 3 times in various conferences. She is a passionate teacher and is very popular amongst her students. Now I invite Dr. Seema Nimbarte to deliver her talk. Over to Dr. Seema Nimbarte, please.
Thank you very much, madam. Thank you so much for such a sweet introduction. And I have always admired Kulkarni madam as a teacher, and I always try to follow her being a teacher. So she is my guru, both way, in my personal life also and as a student also. Okay, so shall I start?
Yes, madam, you can. Please go ahead.
I think this presentation is visible.
Visible. Your screen is visible. You can start, madam.
Madam, I will just make it a full screen view. And
Yes.
just let me know if I change the slide, is it changing on your screen also? So I made it full screen. So is it changing? Or only the first slide is visible?
Just a minute, please.
Yeah.
Just a minute, please.
Yeah, I'll go back. This is the first slide. The topic of today's lecture is PCR, an effective diagnostic tool in molecular biology. So this is the first slide. So I'm changing the slide. Is it visible, madam? Second slide? Third slide? No?
No, madam. No,
Okay,
no, it is not visible.
Okay. This is visible, I hope, right?
Yes.
Aata se change now?
This time, now we can see the change.
Yes. This is a problem. Okay. So, good morning, dear students. Am I audible to you?
Yes, you are very much audible.
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, dear students. So we'll just begin with PCR, which is an effective diagnostic tool in molecular biology. See, basically, PCR, the word PCR stands for polymerase chain reaction. Okay. So here we are going to polymerize a DNA molecule, and it's a chain reaction. It will continue with 1 cycle, 2 cycles, 3 cycles. The number of molecules, DNA molecules will go on increasing. Now, basically, this is used for research purpose. This is not a diagnostic test. But all of you know that since last 2 years, all of us are hearing that Rtpcr test is done for the detection of COVID. Now, since there is no such diagnostic test available for detection of presence of coronavirus in the body of a patient, so this technique was used for diagnosis. As such, this is not a diagnostic technique. Basically, polymerase chain reaction is used for amplification of DNA without cloning it. See, you'll be studying gene cloning in your semester 6 syllabus. If some of the students are of BSc final year, they'll be studying gene cloning. But without cloning, without gene cloning, without using any microorganism to clone a gene, we can amplify a DNA molecule. We can amplify a specific gene. And this technique is the most widely used or versatile technique used for amplification of DNA basically. So first we will study PCR, what is basic PCR, and then we will go to Rtpcr, that is used for detection of COVID. Okay, so I think this is visible. The in vitro amplification. In vitro means in lab, in glass. Okay, so in vitro amplification of DNA by repeated cycles of what cycles are repeated? Strand separation and polymerization. Strand separation means what? Basically, we are going to have a sample DNA which is double stranded. The two strands will be separated. That means the the two strands which are separated will behave as two DNA templates, and then new strands will be synthesized on the DNA template. That means basically when you have two DNA strands, they will be separated and 4 new strands will be synthesized. So doubling of DNA will take place. So in vitro amplification of DNA by repeated cycles of strand separation and polymerization by using DNA polymerase activity is known as polymerase chain reaction. It was invented by Karimulis in the year 1985, or rather we can say the credit of discovery, invention of PCR goes to Karimulis. Now, in each polymerization cycle, as soon as the term polymerization comes to our mind, it means doubling, increasing, okay? So polymerization, the number of DNA fragments are doubled in each cycle, right? Always remember, the number of DNA fragments are doubled in each cycle during PCR reaction. And within a very short period of time, millions or billions of copies of a single DNA can be obtained. Okay, this technique is basically, as I said, is not a diagnostic technique. It is basically used for amplification of DNA. Now, suppose all of you know DNA fingerprinting. Aap sabko pata hai, right? Now, if some crime takes place, then from the crime spot, the accused and victim samples are collected. Sometimes the sample may be just a hair sample, sometimes body scrapings and all. So if you want to identify the accused as well as the victim, we have to match their DNA. Okay, and sometimes the sample that is obtained from the site of crime is so little that we have to amplify the DNA. We need to increase the quantity of the DNA of both the accused and the criminal, right? So in that case, PCR technique is used to amplify the sample of DNA of both the accused and the victim, right? So like that, PCR technique is used in various, you can say, cases for amplifying the DNA, right? So it can be used for making large amount of any DNA sequence. Every minute quantity of DNA can be duplicated to give a large and specific DNA sequences, and hence PCR is a very powerful tool for amplification of DNA, that too without cloning. Okay, now what are the various reaction components that are required during PCR? Number one, of course, the sample, that is the DNA template. The primers, you have studied replication. So for replication, to start replication, DNA polymerase actually requires a primer. So primers are required. All these reactions are taking place in vitro, or will be carried out in vitro. So we need to arrange all these reaction components first. The DNA template or the sample DNA, the primers, the enzyme that is DNA polymerase, then DNTPs, deoxyribonucleotide triphosphates. To synthesize a new strand, okay, we require DNTPs. Like if on your DNA template you have adenine, then adenine pairs with what? Thymine. So we require thymine to be given, right? And if there is guanine on your template strand, then cytosine is required. So for the synthesis of new strands, DNTPs are required. Magnesium, divalent magnesium ions catalyze the reaction, so they are required, and certain buffers are required. Now, so the first requirement is the DNA template. It may be the sample rather, okay? So the DNA containing the region to be sequenced, okay, or the sample containing the region that you want to amplify. Okay, so we need a target DNA sequence which is to be amplified. Size of the target to be amplified up to 3 kb. It should be at least 3 kb can be amplified. Now, secondly, we require primers, right? Second requirement for PCR reaction. 2 sets of primers are required, forward primer and reverse primer. Okay, so on one strand, forward primer will be used, and on another DNA template, reverse primer will be used. Now, primers are generally 20 to 30 nucleotides long, and they are synthetically produced. The primers are complementary to 3' end of the target DNA. The target DNA to be amplified has 5' to 3' end, and primers are complementary to 3' end. That means they attach at the 3' end of the target DNA or template DNA. And the primers are not complementary to each other. If the primers are complementary to each other, they themselves will hybridize. Okay, so they are not complementary. So you should remember one thing: we require 2 sets of primer, forward primer and reverse primer, which are complementary to 3' end of the target DNA. Okay, the third thing which we require is Taq polymerase. In the first line, we have written Taq polymerase. Now, before going to what Taq polymerase is, okay, see, in case of PCR reaction, the whole PCR reaction is divided into 3 steps. Step number 1, denaturation. Step number 2, annealing of primers. Step number 3, polymerization. Now, what happens during the first step of denaturation? The temperature required is very high. 90 to 95 degrees Celsius temperature is required. Okay, and in the third step, where we use polymerase enzyme, usually when the normal DNA polymerase enzyme was used, at the first step itself, when the temperature was raised to 90 to 95 degrees Celsius, the polymerase enzyme loses its activity. It gets denatured. So initially, when the enzyme known as Taq polymerase was not known to us, was not available, the PCR cycles were not very continuous. Scientists had to carry step 1, step 2 first, then take out the reaction mixture, add polymerase enzyme, and carry out the third step. At the end of the third step, cycle 1 is completed. Then they again used to carry out step 1, step 2 for cycle 2, and then remove the reaction mixture and add polymerase. Why the cycles were discontinuous earlier? Because the polymerase enzyme is destroyed at the temperature used in step number 1. Okay, but with the invention, rather you can say with the discovery of Taq polymerase enzyme, all these problems were overcome, right? Now, earlier, E. coli DNA polymerase was originally used in PCR. However, that enzyme is destroyed at a temperature of 95 degrees Celsius, which is required in step number 1, that is DNA denaturation, where the 2 strands of DNA are separated. This enzyme, therefore, had to be added during each cycle of PCR. So we have to take out the reaction mixture from the machine, add enzyme, carry out step 3 of cycle 1. Again, in the second cycle, take out and go on adding. So this problem was overcome. How? In 1988, Sakai et al. Made a major breakthrough by using a heat-stable Taq DNA polymerase. Now, why the name Taq? In the word Taq, TAQ Taq stand for the name of the organism from which this heat stable polymerase was isolated. Okay, so this heat stable polymerase, Taq polymerase, was isolated from a bacterium, Thermus aquaticus. So T of Taq stands for Thermus, the name of the genus of the organism, and AQ stands for aquaticus. Thermus aquaticus. This organism was isolated from hot springs. Okay, this organism lives in water or survives in water. Rather, it is found in water having a temperature of 75 degrees Celsius. Okay. So the optimum temperature for Taq polymerase is 72, but the enzyme remains stable at 95 degrees Celsius also. Okay, and this enzyme has 5' to 3' exonuclease activity. So this enzyme is the best suited enzyme for carrying out PCR reaction. Okay, so with Taq polymerase, you can say the whole PCR technique got innovated. Okay. Okay. What are the advantages of using Taq DNA polymerases? It gives you a higher yield. There is no need of repeated addition of the enzyme for each cycle. We don't have to go on removing the reaction mixture, add enzyme for every cycle. No need. In the beginning itself, you add Taq polymerase and give the machine the order how many cycles you want to carry out. 10 cycle, 20 cycle, 30 cycle. And you don't need to remove the reaction mixture continuously from the machine. Just add the enzyme will remain stable over all, the number of cycles you've ordered the machine to carry out. Nothing happens to the enzyme. So enzyme has very high specificity. There is no need for repeated addition. Okay. And long DNA fragments up to 10 kb also can be amplified. Instead, when we used E. coli DNA polymerase, 400 base pairs were only amplified. But with this enzyme, 10 kb long DNA fragments can also be amplified. Okay. There is another polymerase. If someone asks you what is other than Taq DNA polymerase, do you know any other thermostable polymerase? There is one more, PFU DNA polymerase. Okay, it is also a thermostable polymerase which is isolated from organism Pyrococcus furiosus. Okay, but mostly the enzyme Taq DNA polymerase is used for carrying out polymerization reaction. So what do we require? Number one, the first thing which we require is the machine, PCR machine. It is called as thermocycler. Okay, thermally controlled. Every cycle of PCR has 3 steps. All the 3 steps are carried out at different temperature. So the temperature of each step of each cycle is controlled by the machine. The machine is ordered in the beginning itself. Like step 1 will have how much temperature, step 2 will have how much temperature, and step 3 will have how much temperature. Everything is set up into the machine, ordered to the machine, and the machine is ordered you to carry out these many cycles. And the end, a beep will be given by the machine. Yes, all the cycles are over, the DNA is amplified. So the machine that is used is called as a thermocycler, number one. Number two, as I said, you require sample DNA, target DNA, DNA template. Number 3, DNTPs, magnesium ion, Taq polymerase. All these are the requirements. Okay, so every PCR cycle has 3 steps, as I said. Number 1, DNA denaturation, which is generally done above 90 degrees at 95 degrees Celsius. Number 2, annealing of primer, also called as primer hybridization, which is done at 40 to 50 degrees Celsius. That depends upon different kits or manufacturers who supply you the product, their product. So generally carried out at a temperature range of 40 to 50 degrees Celsius. Step number 3, polymerization, or you can say synthesis of new DNA strand, also called as primer extension. It is carried out by using polymerase enzyme, Taq polymerase, and it is done at 72 degrees Celsius. Okay, so all the 3 steps of PCR cycle have different temperature. Step 1, DNA denaturation, where the double-stranded DNA is converted into single-stranded, 2 single-stranded strands of DNA are separated. That is DNA denaturation at 95 degrees. Number 2, annealing of primer, 40 to 50 degrees Celsius. Number 3, polymerization or synthesis of new DNA strand, also called as primer extension, done at 72 degrees Celsius. And this is done in a machine called as thermocycler. Okay. So this is the basic diagram, you can say. We can repeat 30 to 40 cycles, whatever number of cycles you want, depends upon the sample. If the sample is very less, we have to repeat the number of cycles many times. Okay, and that many times your sample will get amplified. So step 1 is denaturation, which is generally done for a minute. 60 seconds, 1 minute at 94 degrees Celsius, where you can see this is a DNA duplex. Okay, twisted strand, the two strands get separated. Excuse me, that is DNA denaturation. To annealing, as you can see, primers are added, right? So forward and reverse primers are added, and step 3, extension. So DNTPs are added. Say the cycle may be, the third step may be carried out for 1 to 2 minutes at 72 degrees Celsius, and DNTPs are needed, and DNA polymerase enzyme will extend the primer, and 2 strands of DNA are converted into 4 strands. You have 2 copies of double-stranded DNA at the end of 1 cycle. As the number of cycles are repeated, that many number of double-stranded DNA will be obtained. Okay. Now let us discuss the general procedure for amplification of DNA by PCR. Okay, dear students, you are able to hear me now?
Yes, ma'am.
Okay, so what is
Yes,
first
ma'am.
step? First step क्या है? मैंने क्या बताया आपको? First step is DNA denaturation, also called as melting of DNA strands, right? So here, for carrying out DNA amplification, see, if you want multiple copies of DNA, naturally, you need to carry out DNA replication, polymerization करना है. For that, we cannot use a double-stranded DNA. So the double-stranded DNA needs to be, the 2 strands need to be separated. So the first step is DNA denaturation or melting of DNA, because single-stranded DNA is required for the synthesis of new complementary strands during replication. For this, the reaction mixture contains double-stranded DNA. The reaction mixture which contains double-stranded DNA is heated to about 94-95 degrees Celsius for a minute in an automated thermocycler. Okay, the thermocycler machine is automated. So as you start the PCR machine, first step will be DNA melting. Double-stranded DNA will be converted into single-stranded DNA. The hydrogen bonds between the 2 strands are broken. Double-stranded DNA undergoes denaturation, gives you 2 single-stranded DNA molecules. Both the single-stranded DNA now serve as DNA template for the synthesis of new strand. So step 1, melting. Step 2, annealing of primer, attaching the primer, addition of primers. The 2 chemically synthesized oligonucleotide primer, maybe 20 to 30 base pair or 15 to 20 base pair long, are added in the reaction mixture. Primers are added at the 5' end of the DNA template, and in the first step the temperature was 94 or 95 degree. In the second step the temperature is brought down to around 50 degree. Okay, and second step may be carried out for 2 minutes, as long as 2 minutes may be required. Okay, and when you cool down the temperature, the primer will bind to the complementary region flanking the target DNA molecule. The DNA molecule may be very long. Wherever the primer finds complementary region, at that point, the primer will bind to the DNA. This is called, this process of binding of primers to the template DNA is called as annealing, right? Now, temperature and time are the key variables, right? So here, you can say 1.5 to 2 minutes are required for annealing, but annealing is done at a lower temperature, around 50 degrees Celsius. The third step is primer extension, also called as DNA synthesis. Synthesis of new strand will now take place. As in the second step, the primers have attached to the DNA. Now, by the action of Taq polymerase enzyme, new DNA strand will be synthesized. That means the primers will be extended, DNTPs will be added, and polymerization reaction will take place. So deoxyribonucleotide triphosphates and Taq DNA polymerase enzyme are added to the reaction mixture. The mixture, the temperature is heated to around 72 degrees Celsius for 1 or 2 minutes. Okay, it may vary here also. That depends upon whatever product we procure from companies. They tell us how much time will be required for step 3, but minimum 1 to 2 minutes are required. Okay, what happens during this time? When you add Taq polymerase and DNTPs and keep the temperature at 72 degrees Celsius, primer extension will take place and complementary DNA strand, new DNA strands are synthesized on the template. Now, these 3 steps Number 1, DNA denaturation. Number 2, annealing of primer. Number 3, primer extension or DNA synthesis. These 3 steps together make one cycle. And at the end of one cycle, amount of DNA is doubled. Okay. At the end of one cycle, 2 strands are converted into 4 strands. These 3-step cycles are repeated several times. You can repeat 30 cycles, 40 cycles and all, and multiple copies of DNA will be obtained. Okay. This is what you have to study in your syllabus, PCR reaction. Now, okay. So you can see this diagram. You have a template DNA. At the end of the first cycle, this double-stranded, you have duplicated, doubled the amount of DNA. So there were only 2 strands, now 4. At the end of second cycle, so 2 duplexes are converted into 4 duplexes. At the end of the third cycle, again double, 4 duplexes will be converted into 8 duplexes. That means 16 strands of DNA and so on. So there's an exponential amount of amplification, doubling at each step. Okay. So within a very short period of time, even if we consider that for the first step, we require 1 minute. For the second step, we require 2 minutes. And for the third step, we require 2 minutes. That means within 5 minutes, DNA will be duplicated. So within no time, within a very short period of time, okay, you will get amplified DNA. And that amplified DNA can be used for various tests you want to carry out. Okay, you may be having some purpose to amplify your DNA. Your DNA is amplified. The amplified DNA product can be now used for carrying out various detections, whatever you have aimed at, right? So that is about PCR. Now, in general, we'll discuss about the application. PCR is a very specific and accurate technique. So the final DNA product is very pure and identical. It has no mixture and all. It's very pure DNA. It is adapted to amplify DNA, even if it is available in a small amount. What are the various applications? It is used to propagate DNA for gene manipulation, construction of gene library. Okay, that means for all molecular biology techniques, genetic engineering experiments, recombinant DNA technology experiments. Okay, so if you need to amplify your particular gene, that PCR is the basic technique used. You don't have to clone and all. It is used to amplify DNA fragment isolated from organism. Amplified DNAs are used in clinical diagnosis of genetic disease, pathogenic diseases and all. PCR is employed for amplification of DNA to detect criminals, as I said, in forensic science. We have some small samples from the crime site, and the amount of DNA may be so small that it cannot be used for any diagnostic purpose. That's why we use PCR first to amplify the DNA obtained from the crime site, and then we proceed for other tests. Okay. So again, where there is paternal disputes, we go for DNA fingerprinting. Whatever, there is any paternal dispute that baby belongs to which father or which mother and all. If there is any paternal dispute, again, we have to go for DNA fingerprinting. And in that case also, we need to amplify the DNA first. So PCR is used for amplification of DNA for determination of, resolving the paternity dispute or determination of sex of a baby and all. Then we have random PCR, Rtpcr, inverse PCR, single-sided PCR. All these are used for cloning, amplification of DNA. That's the only thing we need to do. Now, let us discuss about Rtpcr in coronavirus testing. Now, before I proceed, I will stop sharing this slide and I'll show you a small video. Just let me know you can hear it or not. Okay, how the reaction is done. As I told you, when we are having Taq polymerase enzyme, it is a single step reaction. That means we have a reaction tube. In the reaction tube, we add the target DNA or the sample DNA. We add the DNTPs. We add the Taq polymerase. We add the buffer, everything, and put this tube into the PCR machine and set up the PCR machine. Okay, there are all, setup. You have to set up. Step 1, what degree? Step 2, what degree? Step 3, what degree? That you will set up in the machine. Then you will order the machine how many cycles you have to carry out. And at the end, you get the amplified DNA. This is not the case with Rtpcr that is used in corona. It's a slight difference. So we'll come to that. Before that, I will show you a video. So I'll stop sharing this. If you have any difficulty in what I told you till now, just let me know. So I will try to share another video. Let me see if I'm able to. Nahi this. Ruma madam. Hello.
Hello, Ruma madam.
Yes, ma'am. Yes, ma'am.
Madam, can we show it from YouTube? I have a YouTube video small. So that I wanted to show the children.
Yes, ma'am. You just share your screen and then open the YouTube channel, and then you can show that particular video.
So I am not able to see the. I mean, Google का screen नहीं दिख रहा है. एक minute, I'll just check.
Chrome open कराना.
Is this?
Now we are going to discuss about
Actually, तुमची screen share नहीं है सध्या.
Okay, madam. Okay.
Amplification of DNA. Suppose we have a sample. We have to isolate the DNA from the sample, perform polymerase reaction, and produce multiple copies of the target DNA. Now, the machine in which the DNA is amplified is called as a thermocycler or thermal cycler, right? So we have an example of tube in which the target DNA is placed. We have to add DSC, we have to add polymerase enzyme inside the tube. So what is the tube contain? The sample DNA, DNTP, polymerase enzyme, and we have to place the tube in the thermocycler machine. What happens inside the thermocycler machine? The target DNA is first unwound. Okay, that means that is step 1, denaturation or melting of DNA, which is carried out at 94 to 95 degrees Celsius. The double-stranded DNA is unwound, hydrogen bonds are broken and converted into single-stranded DNA. Okay, then comes the second step, that is annealing. Annealing means attachment of primer. So we have two primers, forward primer and reverse primer. We have to reduce the temperature to 55 to 65 degrees Celsius and attach the primer. Then comes the third step of extension, which is done at 72 degrees Celsius for 1 to 2 minutes. Where polymerase enzyme, you can see a polymerase traveling and converting the single-stranded DNA into a double-stranded DNA. So at the end of cycle 1, we have 4 strands. 2 strands have been converted into 4. Okay, now cycle 2 starts, right? Again, annealing of primer can be seen, extension of primer, okay, DNTPs are added, and you can see the polymerase enzyme moving, and the dark polymerase and synthesizing double-stranded DNA. So now at the end of cycle 2, we have 8 strands, okay. Now again cycle 3, step 1, conversion of double-stranded into single, step 2, annealing of primer, step 3, attachment of polymerase and converting the single-stranded DNA into double-stranded. So at the end of each cycle, the DNA gets duplicated and the number of cycle continues. The target DNA gets multiplied. The green color is in the target DNA. Out of the total DNA, that is the target DNA which was amplified. Okay. So as the number of cycles increase, the target DNA which we want to amplify also goes on increasing. So you can see here the number of cycles are increasing and the copies of target DNA that are produced shown in green color are also increasing. Okay. So within say 3 to 4 hours, multiple copies of DNA can be synthesized. Okay. So from one single DNA molecule, so many copies of DNA can be synthesized in a very short period of time. This is a machine called as thermocycler machine or Pcr. Now we can check out open the tube and check whether it has been completely amplified or
Okay, ma'am. So, dear students, was that visible to you? Could understand what it is?
Yes मैम. मैम
Yes,
एक बार फिर से दिखाइए ना.
अच्छा फिर समय हो जाएगा, मुझे RT-PCR भी बताना है. कुलकर्णी मैडम, what to do?
बता दीजिए, please show them.
Okay. So you want me to share that thing again, no?
हाँ.
Okay, I will.
Students, now we are going to discuss about polymerase chain reaction. What is polymerase chain reaction? Amplification of DNA. Suppose we have a sample. We have to isolate the DNA from the sample, perform polymerase chain reaction, and produce multiple copies of the target DNA. Now, the machine in which the DNA is amplified is called as a thermocycler or thermal cycler, right? So we have a different tube in which the target DNA is placed, which needs to be amplified. You have to add DSC, you have to add polymerase enzyme inside the tube. So what is the tube contain? Sample DNA, DNTP, polymerase enzyme, and we have to place the tube in the thermocycler machine. What happens inside the thermocycler machine? The target DNA is first unwound. Okay, that means that is step 1, denaturation or melting of DNA, which is carried out at 94 to 95 degrees Celsius. The double-stranded DNA is unwound, hydrogen bonds are broken and converted into single-stranded DNA. Okay, then comes the second step, that is annealing. Annealing means attachment of primer. So we have two primers, forward primer and reverse primer. We have to reduce the temperature to 55 to 65 degrees Celsius and attach the primer. Then comes the third step of extension, which is done at 72 degrees Celsius for 1 to 2 minutes. Then polymerase enzyme. You can see a polymerase traveling and converting the single-stranded DNA into a double-stranded DNA. So at the end of cycle 1, we have 4 strands. 2 strands have been converted into 4. Okay, now cycle 2 starts, right? Again, annealing of primer can be seen. Extension of primer. Okay, DNTPs are added. And you can see the polymerase enzyme moving and duck polymerase and synthesizing double-stranded DNA. So now at the end of cycle 2, we have 8 strands. Okay. Now again cycle 3. Step 1, conversion of double-stranded into single. Step 2, annealing of primer. Step 3, attachment of polymerase and converting the single-stranded DNA into double-stranded. So at the end of each cycle, the DNA gets duplicated and the number of cycle continues. The target DNA gets multiplied. The green color is in the target DNA. Out of the total DNA, that is the target DNA which was amplified. Okay. So as the number of cycles increase, the target DNA which we want to amplify also goes on increasing. So you can see here the number of cycles are increasing. And the copies of target DNA that are produced, shown in green color, are also increasing. Okay, so within say 3 to 4 hours, multiple copies of DNA can be synthesized. Okay, so from one single DNA molecule, so many copies of DNA can be synthesized in a very short period of time. Now take out the window tube and check whether it has been completely amplified or not.
Okay, dear students, thank you so much. So shall I continue with now RT-PCR?
Yes.
Hello, madam. I'll continue with RT-PCR.
Yes, madam. Yes, yes.
Okay. Now see, RT-PCR test is that is done in coronavirus testing. It is reverse transcriptase, or you can say reverse transcription polymerase chain reaction. The word RT stands for reverse transcription polymerase chain reaction. It is also called as real-time reverse transcription polymerase chain reaction. Here, the name real time because it within the reaction mixture, if the patient is infected with coronavirus, then the target DNA from the coronavirus gets amplified, and you can see that amplification because in here, in the reaction mixture, a fluorescent dye is present. The fluorescent dye attaches to the target DNA that was obtained from coronavirus, and that is why the intensity of fluorescence goes on increasing as the target DNA goes on amplifying. So with the amplification of the target DNA, the fluorescence goes on increasing, and you can see amplification in situ at the site. Along with the machine, there is another machine, computer machine, showing you the graph. Okay, so you can see real amplification on spot, okay, with the increase in fluorescence. That's why the name real-time PCR. Now, why reverse transcriptase? Why the name reverse transcriptase PCR? We will come to that. First of all, for COVID-19 testing, usually the test begins with collection of swab, that is from nose or throat. So nasopharyngeal or oropharyngeal swabs are collected. Saliva, mucus or fluid from patient lungs can also be used for tests. So, but basically all over the world, nasopharyngeal and oropharyngeal swabs are collected. Right. So you can see here a patient. Okay, this is a swab. So there is a stick, all sterile, having a swab at one end. It is put into the nose. One swab is put into the nose, nasopharyngeal, and another swab is put into the throat. Okay, so oropharyngeal. Both the swabs are then inserted into a tube containing a transport medium. Okay, so first sample, a swab of a person with COVID-19 is collected. It is put into a transport medium, all sterile, in the tube, and then the sample is taken to the lab. If the sample is collected from the patient's home or from the hospital, it is taken to the lab, or the patient can go to the laboratory also, and the sample will be collected. Now, the swab of the person with COVID-19 will contain mixture of human cells, of course, right? But along with that, virus particles. If the patient is infected with coronavirus, virus particles will also be present on the swab. Then, see, why RT? Why we are using the term RT or reverse transcriptase? Now, in almost all the organisms, the genetic material is the DNA. Okay, the you can say genetic material responsible for hereditary transmission is DNA. But there are certain viruses where DNA is absent and the genetic material is RNA. So look into this. However, so most of the organisms contain DNA as their genetic material. However, the COVID-19 virus, SARS-CoV virus, or other viruses like HIV virus have RNA as their genetic material, right? So if you have viral particles in the sample, they contain RNA. They do not contain DNA. So for PCR, we require double-stranded DNA, okay, because we want to amplify the gene. Right. So this RNA needs to be first converted into DNA. Okay. So listen here now. RNA is chemically very similar to DNA, but RNA molecule, as you can see, the lower one, single-stranded, DNA molecule, double-stranded. In DNA, what are the nitrogenous bases that are present? ATGC. Okay. Adenine pairs with thymine or vice versa. Guanine pairs with cytosine or vice versa. In case of RNA, however, instead of thymine, uracil is present. So what are the bases? AU pairing and GC pairing, right? Now, so this is the structure of coronavirus. The virus is surrounded by an envelope. The envelope protein, E protein, it is called as E protein, envelope protein. Inside is present, of course, virus is a nucleocapsid, we all know. So there is RNA. You can see a small thread. It's very light. अंदर की तरफ एक thread है. So it contains RNA and nucleocapsid protein. So आप लोगों में से किसी ने किसी कोरोना टेस्टिंग वाले पेशेंट का रिपोर्ट देखा होगा, तो they write their N gene. So what is that N gene that is detected? from infected patient, it is nucleocapsid gene. What is the E gene that is detected? The envelope gene. So various countries have, ordered or designed testings where various type of genes are sequenced for or detected. Whether this gene is present or not is detected when the patient goes to the pathology laboratory, gives his nasopharyngeal and oropharyngeal swab for testing of presence of COVID or not. In that case, either N gene, E gene, then there is RNA dependent RNA polymerase, RDRP gene. So various genes are detected. And in almost all the countries, or rather I can say in India, N gene is the most important gene that is detected from corona patients. Okay, so that is nucleocapsid. RNA, envelope. There's so many other genes. So this is the actual clear picture. Envelope having all spike proteins, also called as glycoproteins, envelope proteins. Inside is a RNA and nucleocapsid. Okay. Other proteins are embedded on the envelope. So envelope proteins are also present. Okay. Now the objective of COVID-19 testing is to identify. So the viral DNA may be very long. We don't check over the whole DNA. Out of the whole viral genome, sorry, not DNA, RNA, out of the viral genome, in the patient sample, we detect any one of the gene. The most important is the N gene. Okay, that is nucleocapsid gene. Okay, you can see this arrow-like blue diagram showing various genes. So this is N gene, M gene, envelope gene, spike protein gene. So there are so many genes. N gene is supposed to be the most important one that is to be detected. Okay. So now, now there is not enough viral DNA to detect directly in the patient. Okay. So viral DNA, viral RNA is not in enough quantity for detection. So we need to amplify it. So the first thing before going for amplification, we need to convert the RNA into DNA. Now, all of you must have heard protein synthesis. In protein synthesis, what happens? Or what is the actual normal sequence of synthesis? RNA is synthesized on a DNA template. Okay, so on a DNA template, RNA is synthesized. That is called as a process of transcription, right? That is called as transcription. That means synthesis of RNA on DNA. Now here, we don't have DNA, we have RNA. So we have to carry out a reverse process. That means synthesizing DNA on RNA template. That's why the name reverse transcription. Simple. So here we don't have a DNA, we have an RNA, and we are synthesizing a DNA on RNA. We are going opposite or reverse of the original reaction that takes place naturally. So the name reverse transcription, and the enzyme that is used for this process is called as reverse transcriptase enzyme, RNA dependent DNA polymerase, reverse transcriptase enzyme. Okay. So a process called reverse transcription, polymerase chain reaction, Rtpcr, amplifies many copies of the segment of the N gene. We have taken example of N gene. In various countries other than N gene, other genes are also detected. Say for example, we want to detect the N gene here. So first of all, short single-stranded pieces of DNA called as primer recognize the unique RNA sequence within the viral genome that bracket the target gene, and the DNA primers attached to the RNA template. Okay, so you can see here the DNA primer has attached here. Then comes into action the reverse transcriptase enzyme. Okay, the reverse transcriptase synthesizes single-stranded DNA copy of the viral RNA. That means a complementary DNA is synthesized on the viral RNA by the enzyme reverse transcriptase. Okay, so a complementary DNA is synthesized on RNA template. After that, RNA is removed. Now we don't need the RNA. Okay, we have synthesized a complementary DNA on the RNA. Now the RNA is removed and the single-stranded complementary DNA is converted into double-stranded DNA by RNA DNA polymerase enzyme, right? Here, which polymerase will come into action? The Taq polymerase enzyme. After the RNA is removed, second primer binds to the other side of the single-stranded DNA, and single-stranded complementary DNA is converted into double-stranded DNA by the action of Taq DNA polymerase enzyme. So you have a complementary DNA here, then a DNA primer attaches to it, and we have shown blue color, a symbolic type of DNA Taq polymerase enzyme. It will convert the single-stranded complementary DNA into a double-stranded DNA. The DNA copy then undergoes successive round of cycles. So what you have here, what you can see in this picture is the double-stranded DNA. Okay, made from N gene or made from any other target gene that the company wants to check or the doctors want to check. Okay, so this is the DNA now. Basically, we had the virus having RNA. The RNA, by the action of reverse transcriptase, was converted into, or single-stranded DNA was, complementary DNA was synthesized on RNA template. Then the RNA template is hydrolyzed and removed. You have a single-stranded DNA. The single-stranded DNA is then converted into a double-stranded DNA. This is now your target DNA. Here starts now your PCR reaction. This target DNA will undergo n number of cycles, many cycles. Every cycle will have 3 steps, as I told you. Number 1, DNA denaturation. Number 2, annealing of primers. And number 3, okay, complementary DNA synthesis or primer extension, jisko hum bolte. So the 3-step cycles will repeat, multiple cycles will be repeated, and this DNA copy will be amplified. Okay, so you can see here, the DNA separates, denatures, annealing of primer. Taq polymerase brings about a primer extension or synthesis of new strands. So cycle 1 per 2 strands of DNA are converted into 4 strands. Cycle 2 into 8 strands, and so on. So now the viral genome has been amplified n number of times. That depends upon the number of cycles that you have carried out. Okay. Now, here during RT-PCR for COVID-19, a fluorescent probe, there is a fluorescent dye which is added, and the fluorescent probe allows you to detect the amount of target DNA in real time. Real time, in on spot, you can check how much of target DNA is present. As the cycles will be repeated, the target DNA will be amplified, and you will come to know how many number of cycles of cycles are required for detecting the gene, for detecting the disease. So we've shown here PCR machine. Okay. So these are the, if no virus is present, if the test is negative, the patient is negative for COVID, and the patient is not having any coronavirus, right? Then you get a straight line. Right? There is no peak or no increase in fluorescence. No increase in fluorescence shows that the DNA, target DNA amplification has not taken place. That means test is negative. If you can see a peak in fluorescence, increase in fluorescence, that tells us that target DNA which you want to search, that is COVID DNA, coronavirus DNA is present, right? And as the DNA gets amplified, the fluorescence will also go on increasing. Now, in RT-PCR, there is some value which is written, or that makes sense. Actually, COVID positive or COVID negative is sufficient to get the patient getting treated or for isolation and all. But there is one value there which is called as CT value. Okay, what is a CT value? It is C and T stand for cycle threshold. Okay, the cycle threshold or CT value of an RT-PCR assay refers to the number of cycles that are needed to amplify the viral RNA to reach a detectable level. Got it? So what is the CT value? Number of cycles required to amplify the DNA to a point at which it can be detected. So there is a threshold, and as soon as your DNA is amplified, you carry out cycle 1, 2, 3, 4, 5, the cycle number of cycle go on increasing, and at one point, say for example, after 22 cycles, suddenly you see a green colored fluorescence peak crossing the threshold. That means 21 cycles of PCR were needed to amplify the DNA. Suddenly a fluorescence peak will be seen. In some patient with having more of viruses in their body, very less number of cycles are required to reach the detectable level. So after say 15 cycle or 16 cycles, some patient may show sudden fluorescence. That means 15 cycles were sufficient to detect the virus. Right now, you can read this. As you can see the PPT, you can read this. In real-time PCR assay, positive reaction is detected by accumulation of fluorescent signal. Number one. The cycle threshold is defined as number of cycles required for generating a fluorescent signal to cross the threshold. Number three, CT values are, or CT level rather, are inversely proportional to the number of target nucleic acid. That means lower the CT level, greater is the amount of target molecules present. That means more the virus, less is the CT value. Okay, more the CT value, less is the viral load. Simple. If the viral load is less, more cycles will be required for detection. If viral load is more, less cycles will be required for detection. Okay, so CT value and virus load go inverse, right? Using real-time assay, almost 40 cycles of amplification are carried out. I mean, they wait till 40 cycles whether a fluorescent signal is detected or not. So you can see viral load. If we plot viral load on x-axis and the fluorescence on y-axis, then we can see that after exposure, the viral load goes on increasing. When the patient is having severe symptoms, it is higher and then starts lowering as the patient gets, cured, or as the number of days pass, the number of viruses automatically go on decreasing. Now, in India, we'll talk about CT values. So as I told you, what is the CT value? Number of cycles required for amplification to show a detectable fluorescence, right? If the number of cycles required are less than 29 for detection, then positive, the patient is positive. See, actually, I'll tell you what is the case. If number of cycles required are above 30 or above, then in India, why ICMR said that patient showing CT value, cycle threshold value of 30 or more should be treated as negative patients. Okay. Now, if the value is below 24, patient is in infective stage, that means viral load is higher and rate of transmission from the patient to others will be higher. More than 24 CT value, the chances of transmission are lower, less of viral load is there. Okay. So if you get, fluorescent signal, even after 30 cycle or 35 cycle, virus is present. Simple. But if it is above 30, then it is, according to ICMR guidelines, the patient should be treated negative because there are very less chances, because viral load is so less that the patient may not be showing any symptoms also, and rate of transmission will be almost negligible. So treated as negative. Okay. Now, as you can see in this diagram, there is a threshold level and you can see background fluorescence and all. And suddenly, here we send strong positive CT of 21. So suddenly, at the end of 21 cycle, a strong peak is generated. That means a strong fluorescent signal is generated. It indicates that after 21 cycles, reaction was positive. That means 21 cycles were required for amplification so as to show a positive reaction. And after 30, you can see, after 30, a small peak is generated. That means viral load is very, very less. Though virus is present, but load is very, very less. And such kind of reports are given. COVID-19 virus, qualitative, quantitative, and all in all. So N1 gene, positive result is positive. N gene, 27. Here you can see ORF gene. This is open reference value, something like that. Okay, and it is 29, it is 27. That means 27 cycles were required, 29 cycles were required. And in this one, you can see the last one, the lowermost one, negative. ORF gene, not seen. N gene, not seen. No signal was generated. No fluorescent signal is generated means COVID virus is absent, coronavirus is absent. So with this, I stop, and thank you very much for listening to me. And if you have any question, let me know. So dear students, are you awake? So nahi gaye na?
Students, do you have any questions? You can ask her. Any questions?
No, ma'am.
No, ma'am.
You were able to understand?
Yes, ma'am.
I hope it was not
Yes,
very
ma'am.
boring, right?
No, ma'am.
Okay.
Okay, there are no
thank you, ma'am.
questions.
Thank you very much.
Thank
Over
you,
to you,
madam, thank you for sharing your knowledge and throwing light on this difficult topic like PCR technology. And very nicely you have explained through video, and I'm sure the students must have understood at least the basics of what PCR is. Now I invite Dr. Pratiksha Pohekar to propose vote of thanks. Over to Pratiksha Pohekar, madam.
Thank you, ma'am, for giving me opportunity to put my gratitude in the form of work. Good afternoon, everyone, to the one and all present here. Honorable Chief Guest, Dr. Seema Nimbarte, ma'am, Assistant Professor, Sevadal Mahila Mahavidyalaya, Nagpur, respected Vice Principal, ma'am, Dr. Archana Kulkarni, ma'am, teachers, and all the dear students. I am Dr. Pratiksha Pohekar, Faculty of Microbiology. It is such an honor for me to get the opportunity to thank you all dignitaries on behalf of the Microbiology Department, Dharampet MPDO Memorial Science College, Nagpur. As we know that the polymerase chain reaction, that is the PCR technique, is is often considered as one of the most important scientific advance in the field of molecular biology. I extend my gratitude to our honorable chief guest, Dr. Seema Nimbarte, ma'am. Ma'am, with your effective presentation, video, and simple word, make this difficult technique easy to understand. Thank you, ma'am. Thank you to take out the time from your busy schedule to grace the event. As special thanks to Dr. Akhilesh Peshwe, sir, Principal of Dharampet MPDO Memorial Science College, Nagpur, for providing immense support to make the webinar successful. I extend my gratitude to Dr. Archana Kulkarni, ma'am, to organize the webinar. Thank you, ma'am. I must thank the Dr. Katoke, ma'am, and organized team. I thanks all the students for showing their interest in the program. Once again, thanks everyone for making this event successful. Thank you, ma'am.
Thank you very much, Pratiksha.
Thank you. Ma'am, the session, the webinar is excellent, ma'am. Actually, कुछ हमें भी doubts थे, वो भी clear हो गए.
Thank you. If you have any doubt, you can ask me other time also.
Yes, ma'am.
Thank you.
Thank you, ma'am.
So shall we leave now, madam? Madam, muted, mic muted.
Muted.